Well, hello, everyone. I'm really glad you could make it. My name is Stella Schiefer, and I'm the president of the MIT Alumni Association here in Switzerland. And I recently read a press release about work done by Eawag, and it was about new ways to analyze wastewater to understand how many people in Switzerland may be affected by corona. And knowing that one of our members, Janet, actually works at Eawag, I thought, wow, we have to jump on this opportunity. And I think more people would love to hear about this particular research that is so important for our current situation, and hopefully also will make our lives easier in the next couple of weeks or months to come. So I'm super excited to start this new series of video talks with the MIT Association. And I'm pleased to have two phenomenal guests, Professor Dr. Janet Herring and Dr. Tim Julian today. So Janet is actually the director of Eawag today. Already since 2007, she has received chemistry degrees from Cornell and Harvard in the past, has a PhD from MIT, then actually started her career at Eawag already as a postdoc, and then came back after a bunch of professor positions back to Eawag as the director. And Tim, he is the leader of the pathogen of the Pathogens and Human Health lab at Eawag, and he has a degree in civil and environmental engineering from Stanford, including a PhD. Then he worked at the Johns Hopkins Bloom School of Public Health and has been a research scientist at Eawag since 2014. I hope I got all of these right. Please correct me if anything here is wrong. But basically, to start it off, I was hoping Janet could introduce with a few words again, what is Eawag, what is the mission, what are the responsibilities of this institute for everyone who may not be as familiar with Eawag as you.
Super. Well, thank you very much, Stella, for the introduction. And it's really a pleasure to have so many MIT alumni on board for this online talk. I think it's particularly appropriate because as Stella mentioned, I started my career at Eawag as a postdoc, and that happened because of connections at MIT. That the Eawag director at the time was a good friend of my PhD advisor, and he offered me a postdoc position. So that was a really fun thing. Of course, when I went back to the States, I was thinking, okay, that's it for Switzerland. But I had an opportunity to come back and start as a director in 2007, and that's been really terrific. One of the fun things about being director is that you get to set directions for the Institute, and an important lever for that is the people we hire. And so in addition to Tim Julian, who will be talking to you in just a few minutes, we also have on board Christoph Ort, who is working on this project as well. And this project really came from work that Christoph had been doing for many years, looking at micropollutants in wastewater. And so you may occasionally have seen reports of illegal drug use in different European cities, and that's a tracking kind of project that Christoph has been very involved with over many years. And extending that to look at pathogens in wastewater was sort of a next step that we were able to take. Eawag has a fairly broad mandate in terms of research. It's pretty much everything to do with water, from fully engineered systems that we'll hear about today, wastewater and drinking water treatment systems, but also everything to aquatic ecology in the mountains and water quality. Some of the work on pesticides has also been in the newspapers recently, and you may have read about that. So we have about 500 people on staff with a total budget of about 70 million Swiss francs a year, and about 25% of that is external project money. But we do also have the opportunity to fund internal projects through the Eawag Directorate, and one of those was at least partly funded is Tim's and Christoph's project that you'll be hearing about. And this is one of the other big advantages of Eawag. We're, for this kind of organization, relatively small, and so we can move quickly if opportunities come up. at the beginning of March, Tim said, you know, we could do this kind of work and try it out with wastewater, but we need a deficit guarantee in case some of our other funding that we've applied for doesn't come through. So we're like, yeah, sure, go try it out. And so you can take some risks. Eawag as a whole can sort of buffer these risks, but individual group leaders can start on projects that they think they have a really exciting opportunity for without necessarily doing all the work beforehand to get external funding. So I have to say I'm also very excited to hear what Tim and perhaps also Christoph have to say today about their project on the COVID-19 and wastewater. Thank you.
Great. Thank you so much for the intro, Janet. And now I want to hand over to Tim, who will actually start off with a few slides, because many of these methods, approaches are obviously maybe easier to understand with some pictures and slides. So Tim will give an intro about what is the approach they're taking and give us some overview.
Oh, sorry, well, Stella, I don't see how to mute myself. Can you do that for me?
All right, I'll do that.
All right.
Well, great. Thanks, Stella and Janet, for the introduction. Can everybody see my screen now? Okay, perfect. So today I'm just going to walk you through briefly some background about the wastewater-based epidemiology for a pandemic we've been working on. And just to follow on what Janet was saying about the flexibility of Eawag and funding, the deficit guarantee was also really nice, but this also built out of relationships that had formed through other flexible funding opportunities. I worked closely with Tamara Cohn and got to know Christoph because of the opportunities available at Eawag to work through flexible funding. So the first thing to say is I'm just one of a large group of people working on this. At Eawag, there's Christoph Ort, who's a PI, as well as research assistants Corolla Benzinger and another group leader, Krista McArdell. Elise Stackler is a postdoc, and Leah Kaduf, who's a research assistant. There's also EPFL through Tamar Cohn, who's another PI, co-PI on the project. And Javi Fernandez-Cassie is the one helping lead the protocol development. And then we're also working with Gilbert Grube. But another thing is there's just a lot of people supporting this too through sample collection and general support for the project, and their names are all listed here. The other thing to say that's really important is that we're just one of many groups who are interested in this, and there's a huge global effort working on wastewater-based epidemiology, WBE. And there's this website, COVID-19 WBEC, if you're interested in looking at more sort of who's involved in this, that shows all of the research groups who are working on it. And today I'm just going to walk in about maybe 5 to 10 minutes, the theory behind what we're working on, some of the methods we're doing. I'll show initial results and then talk about the next steps. And then there'll be time for a structured question and answer session that Stella will help lead. So for theory, the concept that we had is that wastewater might be able to act as an early warning for diseases that are shed through our fecal material. So for example, here's a plot of the observed confirmed cases. So this is the clinical case data you might see for an epidemic. And for the COVID-19 case, this is what we saw in mid-March, is over time, an increase in the observed number of cases. And the idea, part of the idea behind what we want to do is that there's a large fraction of unobserved true cases. And that can happen for two reasons. One is that there's a lag between the first case, the first time someone's infected, and the time that it's observed confirmed clinical case. And that's because symptoms take a while to go before you go to the doctor, and then the test itself takes a little while. But there's also a portion of cases, and that's what you see along the horizontal axis, the difference in time. But you also see a difference in that the observed confirmed cases are only a fraction of the total cases because there's asymptomatic or mild cases. There might be limits with testing kits, especially early on with the new disease. And then if there's a disease like SARS coronavirus 2 is that's shed in fecal material, the idea is that there should be some fecal shedding from the unobserved true cases that makes its way into the wastewater. And people can excrete potentially a lot of copies, genetic copies of material for this virus. So we get a little bit of amplification. Instead of trying to find, 1 or 2 people, we're trying to find 10 to the 7th, 10 to the 8th copies of RNA, potentially in wastewater. So the idea is that there might be some magnitude of effect here that allows us to detect cases that would otherwise go undetected. And then, of course, even though it occurs in wastewater, there's still it takes us some time to detect coronavirus in wastewater. And the main lag is that we want to collect samples throughout the day and pool them. So it takes 1 day to collect the sample, and then it takes about 10 to 12 hours to process and detect. So we still get an early prediction period of anywhere between 5 and 14 days. Relative to what we use now, the observed confirmed cases. And that's driven, like I said, by those things, people's presentation at hospitals and testing lag. So there's the potential to predict earlier disease outbreaks. We're also interested in that part, but the second thing we're interested in is whether or not wastewater can help inform the dynamics of a disease. So for example, we might see a decline in confirmed cases, and then due to seasonal patterns in a disease or changes in public health interventions, we might see a rebound. And so another question we have is whether or not the coronavirus in the wastewater can also detect that rebound. And for that, we'd have to be able to see with relatively high resolution what our sampling would be like. So that's the theory, the general theory behind what we're trying to do. But what we're actually doing is we have a sample collection strategy where we have a number of wastewater treatment plants, including in Ticino, where the outbreak started, where we're collecting wastewater with quite high frequency, at least 3 times a week. And so we started this on February 27th, which was 2 days after the first case was reported in Switzerland, and it was in Ticino. And we started collecting at a lot of the wastewater treatment plants in the area. We also immediately started collecting at Zurich and at Lausanne, which was the location of the wastewater treatment plants closest to the 2 institutes, EPFL and Eawag, most involved. And then what we do is we collect those samples, and what we have to do is we have to first concentrate the samples, which we do using ultrafiltration. So we have our inlet wastewater collection, and then we add 5,6 as a process control. That's a bacteriophage, an enveloped bacteriophage that allows us to estimate how much we recover from the sample. We filter it to remove large materials that might interfere with our concentration step. And then we use an ultrafiltration to concentrate the viral particles. Once we have the viral particles concentrated, we move into a viral nucleic acid extraction method, and this is the same as they would do for clinical diagnosis. And then we go into real-time PCR. And real-time PCR is important because what we're trying to do is to inform dynamics. We need to know the quantity or the concentration of the virus in the material. And although this sounds really straightforward, there's a number of challenges associated with this. And the first one is that we're looking for a very, very low concentration of virus. So if 1 or 2 people are infected and they're shedding up to 10 to the 7th copies of RNA, that's diluted in the entire catchment area's wastewater treatment. So now we drop down to maybe 10 to the 2 or 10 to the 3 copies of virus. So very low copies. And then the other thing is, of course, this is contaminated with a lot of other material. And I show here organics as just one example, but there's a lot of other stuff obviously in there besides the virus. And so we need a large volume, something like 50 mils or 300 mils. And then we have to concentrate that down to a very low volume so that we can detect it. And so through the combination of concentration, through the ultrafiltration, as well as the RNA extraction step, we get that 50 mils down to about 80 microliters. And now we've co-concentrated not only the virus, but also these inhibitory compounds. And these become important for the quantification step. In the quantification step with QRT-PCR, we take a small subset of the 80 microliters, 5 microliters, and we look for virus in the sample. But because we co-concentrated inhibitors, we end up under-detecting. So even though there's 4 virus shown here, it might show 1 or 2 virus, for example. So there's inhibitor interference. So what we've been working on for the last month is trying to develop strategies where we can concentrate the virus without concentrating the inhibitors, or we can come up with different detection methods. For example, qPCR is what we're looking for, but there's been some suggestion through collaborations that something like droplet digital PCR might be better at handling this inhibition. So we're also using that as an approach to improve our quantification methods. Despite all of these challenges, we've still had a number of successes. We have the first results from Switzerland reported. And this is that's Javi shown there. He's a postdoc working on the project, working in a biosafety fume hood with a qPCR step. And so what we see here is a qPCR data, the raw data that you'd expect. And we have the calibration curve, which are the detections of our standard. So we put a standard into the sample in the qPCR, and it fluoresces as PCR duplicates the DNA, or the cDNA. It starts to fluoresce, and there's increasing fluorescence. And we use standards at different concentrations so we can benchmark and estimate our concentrations in our sample. So in green are highlighted the standards, and in red are 2 samples from the Zurich wastewater treatment plant, one on the 19th of April and one on the 29th of February. And so what we see here is the 19th of April, when there were quite a lot of cases in Zurich, we get not only a hit for SARS coronavirus 2, but it's also quantitative because it lies within the standard curve. So we can estimate the concentration, and that's what we want to help us inform dynamics. We also see on the 29th of February, when there are only 6 reported clinical cases, there's a detection of the coronavirus 2 in the wastewater as well. And so what that tells us is that there's something happening. There's people within the catchment area actively shedding the disease. This falls outside of the quantification limit, so we're still working on trying to understand what that means, and also generally trying to understand how these concentrations relate to the number of infected. So another thing we did is we also looked at Lausanne wastewater treatment. And what we see is, again, these quantitative or semi-quantitative concentrations, the RNA per mil wastewater estimate is on the right side of the y-axis, and on the left side is the number of cases or shedders. And what we plotted here was the total number of cases in the canton over time. And this is estimated based on clinical case data times 10, where we assume that 10% of people present as clinical cases. And then we estimate what their shedding pattern would look like based on the amount that's shed in the feces and the duration. So that's the modeled amount. And then we see the RNA that we measured in Lausanne over time. And so you see that it's very, very low for the first 2 dates in March. And on the 3rd date, it stays relatively low, and then it jumps up sort of towards the end, the 12th of April. And so what this shows is that there's some sort of trend, but that, again, it doesn't track perfectly, and there's some issues we're having with the samples, and it all relates back to the combination of the concentration and inhibition, but it might also be natural variation, and these are outstanding questions that we have. But of course, this is a global effort. We're just one group that's working on this in the Swiss context. There's some really comprehensive results from wastewater treatment plants in the Netherlands, where they show basically the same thing. They have data from the number of cases in 2 areas, in Amsterdam and Utrecht. And then they also have the gene copies per milliliter wastewater using different gene targets. So we use the E gene of the SARS coronavirus 2, and they looked at the N1, N2, and N3. And again, they show increasing case numbers with increasing copy numbers. And of course, there's work from MIT on this, which is not surprising. Eric Alm's lab showed that SARS coronavirus 2 titers in wastewater are higher than expected from clinically confirmed cases. So that's also a really interesting preprint that came out. And there's lots of other work on this topic globally. This COVID-19 WBE website shows all of the labs that have published on this through preprints or peer-reviewed publications. So that's also a nice resource to check out. But for our conclusions and next steps, for our work in particularly, we've shown that RNA fragments are detected in Swiss wastewater. And all samples tested to date have fragments, although they might be below the limit of quantification. More information is needed to make this a monitoring tool. How does SARS coronavirus 2 detection in wastewater complement clinical test results is a question, an outstanding question we have. And also we're wondering how do assay limitations, this relationship between detection limit and quantification, influence our interpretation? We're also interested in what are the risks from SARS coronavirus 2 RNA fragments in wastewater. And this parallels to other viruses. Wastewater work requires some level of protection because there's a lot of enteric diseases, antimicrobial resistance, and so we need to be protected when we interact with it, of course. But for SARS coronavirus 2, there's no known transmission from this route. And there's very limited reports on the infectivity of SARS coronavirus 2 in feces and also wastewater. And so this is a huge sort of outstanding question that still we're waiting to see kind of what comes out for that. And there are a lot of other resources. There's just a few here related to facts about COVID-19 virus transmission through water and wastewater generally that I encourage you to check out. The last thing to say is we've received a lot of support, not only from Eawag through the deficit guarantee, also EPFL has given us some support. We just found out that we were successful for our Swiss National Science Foundation. We've been approached by the Swiss federal government for help, financial support too for this, as well as the city of Lausanne and a number of utilities. So that's all I had for this portion. And now I think we go to the structured questions.
Okay, great. Well, thank you, Tim, for this comprehensive overview. And I will just kick off with a few questions on my part, but as I also shared with you in the invitation, there's actually a digital Q&A, so feel free to also put your question there that we'll go over after. I'll just start with some questions I had noted down. I was curious to understand, so how many samples would I actually need to make meaningful predictions given what you've learned so far? So you're only using a few water waste plants right now to get samples. How many samples and how many treatment facilities would I have to cooperate with to make really good predictions? What do you think?
That's a really good question. And I invite Christoph too to also jump in anytime that he wants. But so I think there's two general goals for this type of work. And the first, where I think it'd be really valuable is early detection of an emerging disease or a reemergence of a disease. And for that, you'd really need regular sampling across the areas that you're concerned it will move into, the geographic spread. So every few days, every couple days at least. And for example, we collected early in late February for this in Ticino and Zurich and Lausanne specifically so that we could try to look and see how feasible this is once we get our methods developed. For predicting trends, we really think we require high resolution time series. So right now we're collecting 3 days a week at each of the wastewater treatment plants. And we'll see once we have the data if that's enough or if the trends really shift sort of at lower frequency or higher frequency than that. So I think I don't have an exact number, but I think the areas you're interested in with high frequency sampling is something that's probably needed.
I think what we could add at this stage is our collaborator Gilbert Groib from the hospitals in the canton of Vaudois in Western Switzerland. I think he said they analyzed up to 1,200 samples per day at the peak. And we have around 700 to 800 treatment plants in Switzerland. So instead of sampling for individuals, if you went for, let's say, we now targeted in an early warning system in correlation to COVID-19, we watched how many 19 treatment plants, if we spread them well over the country and take the biggest ones, we can cover a population of 2.5 million people. And in the 12 treatment plants with the small ones in Ticino that we sampled to get a high geographic resolution, we covered around almost 1 million people. And we've also taken the treatment plant that includes Zurich Airport to see whether or not the signal is there earlier or not. But it's clear if you want to cover all of Switzerland, you have to sample all the 700 to 800 treatment plants.
That maybe actually leads to my next question. I guess I was wondering, should this be a mandatory or standard analysis that is just conducted in every wastewater treatment plant?
Yeah, so I think that we first need to prove that this can work and that the information it provides is useful. I think that's sort of the first step. And that I think once we show, if we can show that early detection is really important, then we need responsive networks of trained staff that could be mobilized to start this type of effort in the future. And so for our SNF, which was funded, we have a portion of it working with staff at wastewater treatment facilities to build up that network in Switzerland. Because I think it's very, very useful in a responsive way to the emerging diseases.
Mm-hmm. And right now, it's fairly obvious that you need a lot of lab equipment to make the analysis, to shrink down 50 mil to the highly concentrated small portion. But I wonder, if this proves successful, is there any chance that this could be a more standardized and scalable analysis approach you could share with any end consumer? Could I just go to the loo and try to figure it out if I have corona with my own sample? Would that be a suitable test to share with everyone?
That's a good question. Yeah, so the equipment we need is not state of the art. The supplies are generally available at research institutes and hospitals, but not generally in people's homes, centrifuges, qPCR machines. The other thing to note is for fecal processing, not everyone excretes the virus. Not everyone who's infected excretes the virus in their feces. So there'd be a relatively high false negative rate. And the detection techniques for presence absence would be better applied to other clinical samples, for example, a nasal swab or something like that. So I think if it becomes available in your home, it would be through a different exposure transmission route or sample type. Yeah.
Right. Then I'm wondering also, so you already said Switzerland has promised you a few funds going forward. Do you think in the past, the Swiss government or other foundations have invested enough in this research proactively to make these type of methodologies available?
Yeah, so that's a good question. So I think we're getting a lot of support for this project. I think for pandemic response, it was not really something, I don't know, it wasn't something that was really clearly on everyone's radar that they needed to be invested in. But that said, I mean, there's a lot of other investments in this type of monitoring. Tamar Cohn's looking at other infectious diseases, and Christoph Ort has worked extensively on other public health relevant issues like illicit drug use. Christoph, do you have other things to add?
No, I think if we look back 10 years ago, I mean, now it's just a terminology, wastewater-based epidemiology, and I think now it really becomes, or it deserves what the name stands for. Before that, many people, both in chemistry and microbiology, have done wastewater-based epidemiology. It was just not termed, or it was just not named this way. And 10 years ago in Australia, yeah, people laughed when we said, or when my colleague said they will analyze illicit drugs, and now it's become a routine tool to monitor illicit drugs and other questions through wastewater. I think, as we have seen this now as a global threat or a global issue, and with the knowledge that we have gathered, both logistically as well as with regard to how to communicate results and how to evaluate what the big sample out of a wastewater means rather than a clinical sample, we've probably got quite some head start and maybe also more credibility, so that it's not a new method. It's just now applied in this context, and I hope that this will catch up more quickly than in the past.
Mm-hmm. Yeah, what you just mentioned. So this method has already been used in for previous types of diseases, correct? For example, I believe there has been monitoring for polio and other things via wastewater treatments. So is there anything new about your approach, or are you basically just trying to come up with new ways to properly calibrate the models and understand how many people are really affected by this particular type of disease?
Yeah, I think a lot of the early work on, for example, hepatitis A or norovirus or poliovirus. For poliovirus, that was really focused on areas, particularly low-income areas, where clinical case definition, identification of polio cases was poor or insufficient. And so it was a really nice complementary approach to try to track down these cases. And I think that links really closely to what we were trying to do with the first of the two goals I talked about, which is tracking the geographic spread, identifying areas where it comes new. The second part, looking at seeing if the dynamics in wastewater transmission can help inform the disease outbreak, can contribute to the clinical case understanding of the transmission dynamics, the ebb and the flow of the disease. That I think has a lot of chances to really advance and is a relatively new concept for infectious disease, and as far as I understand.
Okay, I have one more question before I'll open it up to the broader Q&A, and I see there are already a few questions on the digital Q&A. So wastewater treatment is obviously one path forward to try to model the number of infected people. What other methods do you think will be key to detect outbreaks in the future that you would bet on?
Thanks for the question. Yeah, so my main expertise is poop, so that's sort of what I focus on. I think wastewater-based epidemiology is a really promising approach to track the geographic spread. But some other methods that we've talked about that we think are really important are random sampling, and particularly pooled sampling. If you pool samples, you can help reduce costs quite a bit and still maintain some understanding of disease spread. You mentioned self-testing, and that's, I think, something that could be in the future really valuable, particularly if technical expertise is reduced for that types of test. And then other sort of more novel ideas that I think need a lot more traction, need a lot more research, would be things like social network mining to understand what's happening through, for example, self-reported symptoms. Those are just some ideas I'd have.
Great. I'm just starting to present other questions from all of you, so thank you for submitting. And I'll just start at the top. So do you think there's possibility that such kind of tests can be rolled out for individuals? Oh, I think we already had that. I think you already answered that. So I will just go to the next one. This is a kind of early warning system. How should public health administrators interpret the results if they are such relatively high false negative rate?
Oh, that's a good idea. So the high false negative rate comes from the individual. So the idea is that if we test if we test 50 people who are infected, not all of them would be spreading it, so maybe 30 would. And so at a population level, wastewater treatment captures from a whole population. Once you hit a certain threshold percent, once you get past a couple of cases, then your likelihood of having it there is much higher. And so I think the idea would be once you get a detect and you can confirm it, that means the disease has sort of spread. So I think there's those two sort of functions, is that we have to account for that percent that don't have the feces or the RNA in their feces in the models that estimate total population. But that from a population level, the false negative rate should be sort of manageable. I hope that makes sense.
Publius happy. How can I get updates on the research going forward?
Oh, that's a really good question. So I think there's a lot of movement in the field generally through preprints and publications. For us in particular, I don't know, Christoph, do you have ideas on how we update our research?
Well, we communicate as soon as we have more data, but I think the Med Archive with over 3,000 probably in the meantime, like last time when I looked it up, wastewater and coronavirus, and it was 2,700 papers or preprints. Oh, sorry, not corona and wastewater, just corona related. And then as what you mentioned, this COVID, the wastewater-based epidemiology webpage, where the different groups are registered to share and exchange ideas.
You can also share these again afterwards in a follow-up email. I think that would be helpful, maybe.
Yeah.
Do that. Okay. Are you, other institutes, sharing these insights or plan to share these insights with developing countries where I did not see a lot of traction from the COVID map?
Yeah, that's a really great question. So we just started working a little bit with so Eawag also has a department sanitation, solid waste, and water for development that focuses a lot on just general development issues globally. And so we just started talking to them about options for sort of porting this over. But I think it's one of those things where it's a question of sort of I guess it's also partly a question of whether or not these are the right things to be doing for certain areas. For example, we don't know yet how useful this is. And so that's one of the things we talk about is when we interact with different stakeholders is where should the resources be going. So I think that we're interested in this and we're trying to push this forward, but we need to make sure that it's something useful.
Mm-hmm. Next one. So what does your model look like to estimate number of infections based on the virus concentration?
That's a good question, and it's really early stage. The model is really early stage. So right now, what we're thinking of doing is just incorporating the parameters into a general model that relies on the estimates for the concentration. So some of the parameters that we need to include will include things like the percent of people who shed it in their feces, the time course of fecal shedding rates, the population catchment size, factors influencing the wastewater itself, like the flow rate. And so those are just some of the sort of things early on, but there's a lot more work we have to do on the model. Christoph, do you have more to add?
Well, there are different methods with regard to genetic sequencing, but that's all up in the air and really early stage, but there's promising different ways to narrow down the number of cases, which may require more input data, as Tim mentioned, or then more advanced techniques to get mutation rates, if I understand correctly. That's what we do together with other collaborators.
I think the next one's also a very interesting question. Assume a new virus arises. When would it actually be investigated? You mentioned some measurements of COVID-19 in February. So I believe it was February 28th you started taking your measurements. Was this based on COVID-19 observations in China? Put simply, how did you know to investigate it?
That's a really good question, and it kind of goes back to the very start of this project, which was because of the relationships we had developed through Eawag and through the discretionary funds. Tamar Cohn, Christoph, and I just had really informal conversations in late February about what COVID-19 in Italy meant and what we should do, what we could do to prepare for this if it were to come into Switzerland. And so that's why it sort of happened. So I think that now there was some hesitancy early on in the disease because we weren't sure if it would follow the path like the original SARS coronavirus or if it would be something new and turn into a pandemic. But once it moved into Italy, it became clear that this might be something different. And so that's how we knew to investigate it. And that's how we started collecting samples really early, or as soon as there was a case in Switzerland.
And maybe to add, I mean, we use the same primers or the same material that is needed for the quantification in the PCR as is done with clinical tests. Obviously, if the genetic information is not available, then we can't do it with wastewater because then it's not an early warning system, then it's just retrospectively following the epidemic curve. But if the tests are available, if the genetic information is available, then we can do this next time, hopefully even more quickly, because then we'd be ready. We don't have to call treatment plants and explain. And they were very cooperative. I have to say almost all treatment plants we call based on personal relationships or because cantonal authorities, they are so helpful. They really do everything they can. But next time it would be great if we just would have to press the red button, or if we analyze one sample a month or one sample a week. As long as it's 0, 0, 0, we know it's not here. And then we, as Tim explained, we would see from 1 week to the other, oh, now it pops up in one of the 19 or in one of the 100 treatment plans that we regularly analyze in the future, maybe.
Next question is, does the amount of virus signal correspond to the number of infected people? There's probably interpersonal variability of virus shedding.
Yeah, that's a good question. So I think some of the early data, I showed a little bit from the Netherlands, and there's also a preprint from looking at Paris wastewater that shows as the clinical case number that increased, so did the virus signal in the wastewater. And I think early on, that's probably true. As we see log exponential growth, log increases in people, will probably see an increase, a log exponential increase in the virus signal. Whether or not we can use the virus numbers themselves, the concentration we detect to infer the number of infected people is still an outstanding question. And I'm a little bit skeptical of that, but it's something we're really interested in. And then whether or not we can see sort of downturn patterns, that rebound effect I talked about, that's also something that we don't really know because of the length of time people can be shedding RNA fragments in their feces.
Mm-hmm. How do you see the potential of the monitoring tool to help public admin decide whether to implement or ease restrictions, e.g. partial or full lockdown?
Yeah, I think that's a really great question. I think there's potential once this is up and running to be able to show trends in the data in a similar way that we can show trends in the clinical case data. So to see a sort of a leveling off, maybe a little bit earlier than we saw with the clinical cases, and maybe even to see a decline in concentrations or to see that rebound effect. There's potential for that, but that's all work that needs to be done.
Maybe I just inject a question myself here. So once you have a perfect model, Janet, Tim, Christoph, would you all work together to actually make a recommendation to the government how to maybe decide on lockdowns or partial lockdowns based on your data?
So a lot of our structure for what we're trying to do fits in the realm of trying to help inform the epidemiological models. So the Swiss National Task Force for COVID-19 has done a really nice job of coalescing scientists around the issue and including relatively complicated epidemiological models. So I think if we can get our data up and running and our model up and running and make it just open source and accessible, then we're hoping that would be used to help inform those models. So we know what the clinical case data is, which goes into those models, but then also to be able to add this additional input data to say these are the trends that are happening a little bit before we're observing with the clinical case data. So I don't think that we would ever base any recommendations only on our data set. Yeah, that's a good question.
If I could just add to that, I think Tim mentioned the National Task Force, and I think that's been a really very effective mechanism to bring together good scientific input and make it available to government officials. And I think it's very important that scientists work together in that way, as opposed to everybody sort of jumping out and saying, "Hey, use my model, use this, use that," because that just leads to a lot of confusion.
Mm-hmm. And there are, in the meantime, a lot of, well, there is selected cantons who do this really very specifically, and also together with random testing, which would help to see whether or not we can validate our model or we can calibrate our model. And then the Swiss Federal Office for Public Health, they've also been in touch, and they are considering using this among other indicators, the wastewater as an additional indicator.
The next question in my project is, can this testing process result in situation direction where we can also investigate why a high percentage of individuals infected by COVID-19 do not show any symptoms?
I don't think so.
Yeah, probably not.
You ask about why. I don't think we can derive direct causal relationships out of the wastewater. But that's my humble opinion, not knowing all the details about what I mentioned before with these genetic mutations, that with sequencing one could maybe get more information. Tim?
Yeah, I think that's true. I think one of the sort of interesting questions is also, right now we're using wastewater treatment plants, but there's potential to sort of go upstream. I think that's the right word, upstream, and track sort of blocks or communities that might have infections versus others that don't. But I don't think this will ever be at the point where we can track it down to individual households. to say that these people are infected. So I don't think that this would ever be used necessarily to identify asymptomatic people. I think random testing would be sort of random clinical testing would be the approach for identifying asymptomatic people.
It's also the wastewater-based epidemiology approach, due to ethical reasons, has been to keep it as anonymous as possible, been restricted to larger wastewater treatment plants. That's also where we have the most information about the large population efficiently with a single sample. But obviously there have been thoughts and attempts to, now we should, as we open or reopen schools, we should capture or test all effluents of the schools. I mean, this is already technically quite demanding, because in treatment plants, we can rely on their routine sampling, and the intermittent flows and representative sampling at the effluent of individual premises, may it be hospitals or elderly homes or schools, is much more challenging. Obviously, many more points. So what Tim mentioned before, to pool their clinical samples may be more efficient than trying to get a sample from wastewater. But depending on how much money is involved, how much benefit we can gain, maybe there will be apparatus or instruments to better sample for individual or from individual premises.
Next question, Julian is asking, where in the wastewater treatment plant is the sample taken? And are there any challenges to take this specific sample?
Well, this just ties into what I said in my last answer. We take the raw wastewater, maybe after a fine screen or after a grit removal, but it's really the untreated, unfiltered raw wastewater. And in treatment plants, this works quite well. What we don't know yet is how many of the viruses or fragments of RNA are bound to particulate matter, but a lot of this is also in suspension. And related to this, as far as we know, there is not too many challenges, because we've seen that the routine sampling setup of wastewater treatment plants is typically good enough to also capture individual toilet flushes or individual small amounts, or from a small fraction of the population.
Ariane is asking, do you have an idea of what this type of test would cost? Could it deter some people, or would it be infeasible for every wastewater plant to put this in place?
So the estimated costs we have right now is basically, method development. So it's not, at scale. It's not a commercial kit or anything. Would be about 150 to 200 Swiss francs per sample.
Just consumables, right?
Yeah, just consumables. Yeah, plus labor.
Sorry, can you repeat how much was that?
Just consumables at our scale, 150 to 200 Swiss francs per sample. And so I think that this could be really dramatically reduced with once it's at scale, like a commercial kit or something like that. So but I also think that this is really important, right? So this pandemic has had massive economic consequences. And so if something similar to what had happened, I think we could argue that if this is effective, this could be something that would be a worthwhile investment.
And not everyone would need to have a PCR. So you could just prep the samples and then submit them somewhere centrally, I would guess.
Mm-hmm. Chelsea's wondering, are the COVID-19 RNA segments in waste always intact enough for detection?
No, I think that there's the potential. We have a couple of issues that limit our ability to detect it. So it might be present in the wastewater and just be not detectable because of our issues with concentration. The volume we can originally concentrate. So if it's below, about 1 copy per 50 mil, or below 1 copy per 300 mils, then we just can't detect it because we're not concentrating that sample. And then there's also these issues about whether or not the RNA is sufficiently intact. And RNA is really easily destroyed by the environment. RNases are present everywhere. And so we're hoping that we're concentrating the intact virions, the non-infective intact virions, through our process. But certainly, I'm sure there's segments that can't be detected.
Great. There's one more question, Howard, but I'm not sure if that's a question. Considering the US has started opening possibly too early. Maybe it has. But
oh yeah, that's right. Can I just ask if the US is opening early, and they are in some states, can you detect already anything, say, in the state of Georgia or other states that are opening too early?
Yeah, I think that wastewater monitoring could be a useful tool for sort of helping decide whether or not to open and sort of ease restrictions, and then also to monitor the impact of choosing whether or not to ease restrictions.
But certainly from non-detect, from the start for the first outbreak to the first detects, I think we're much more confident that it can really serve as an early warning tool. Whether or not it can really serve as to pick up whether someone opens too early or not depends on many other factors, and on also the variability that we will see hopefully in our day-to-day samples that we will evaluate, and then also how long the people shed. We've seen data that people who were tested positive once, they will also be positive tested 28 up to 28 days or negative after their first positive test. And with this long tail, I'm not sure how quickly or how effectively, how reliably we could pick up early openings or not.
I'm really interested in the rebound problem.
Yeah, absolutely. Yeah, we're excited to see more once we get the method down and we can estimate reliable quantitative estimates. We're eager to see how responsive that is relative to the clinical case data.
Good, thank you. Great.
Well, I think given there were no more questions on the Q&A, I would just like to give a huge thanks to Timothy, Janet, and Christoph for joining and presenting today. Thank you for this very interesting insight into what you're doing. Please keep on doing it because it's obviously important, and hopefully it'll be meaningful, not just for Switzerland, but for everyone in the world. Yeah, and thank you everyone who joined. I hope you found this interesting. Please also feel free to give us feedback on what we can do better, if we do more talks in the future. If you have an idea for a great talk yourself, please feel free to also just to submit it. And we'll make sure to also send some suggestions, suggested links to you afterwards in an email so you can follow up on some reading or some research if you want to. Yeah, thank you so much.
Well, thank you so much for the opportunity.
Bye, everyone. Have a good day.
Bye.
Bye-bye.